Welcome to the opening session of the 2021 MIDD Plus Scientific Conference. One housekeeping note, if you have questions for our speakers at the end of the presentation, please use the Q&A panel on the right-hand side of your dashboard. Time dependent, we will address those questions after the presentation. Now it is my pleasure to introduce Sean O'Connor, CEO of Simulations Plus, and joining him is Brett Howell, Divisional President of DILIsym Services. Sean, welcome to the virtual stage.
Thank you, Arlene. And in advance, I know the conference is going to go perfectly smooth, no Golden Globes type issues with Zoom. And thanks for all your efforts in terms of pulling the conference together here. Welcome, everyone, to the first virtual conference, Model-Informed Drug Development Conference by Simulations Plus. I'm Sean O'Connor, the CEO of Simulations Plus, and Brett Howell will assist me and cover some of our presentation today. Look forward to the talk here and all of the exciting agenda topics that we have over the next couple of days. Appreciate that you've joined and kicked it off with the first session. And without further ado, Brett, let's move a slide. Modeling and simulation in the drug development world. Boy, it's been a overnight success, everyone, right? No, it's been 20 years in the making. Those early days of Lou Shiner and Carl Peck, Larry Lesko, Stuart Beal, our own Dan Weiner and Walt Woltos, bringing ideas of modeling and simulation and how it can impact the drug development process back in those early days 20 years ago. What is it? Can it be used? These were the questions that we encountered. And over the following time frame, those opportunities to impact drug development were found and proven out and expanded over time. And how do I use it? Translated into where can I be impactful in our drug development process? Today, we're flourishing out there, flourishing in the sense of a transition to model-informed drug development. How do I base my drug development process on the tools and capabilities and techniques of modeling and simulation? Today's and tomorrow's conference is meant to celebrate that achievement, that progress that we've made in using modeling and simulation, celebrate that and cover today's issues and opportunities that exist. A few logistics items, 2 days of conference, a lot of sessions, number of panels, and training opportunities from a recruiting perspective. Fill the schedule, as well as some awards and some fun time as well. So we look forward to that. Next slide. I point your attention to the portal, which is your means by which you can navigate all the conference offerings here, whether it's getting to the main stage, which you guys have successfully accomplished here by your presence, but also in terms of finding the other sessions and speaker presentations. How do you access those? We've got a little bit of a party for Bob Clark on the agenda, and then obviously if you have any questions along the way, interest and want to pursue the Simulations Plus products and services, those offerings that we deliver to our clients, you can access that and gain information through the portal there as well. I turn my attention now to, who are we, Simulations Plus, and what do we bring to the table? Next slide, Brett. Drug developers are hit with a barrage of questions in this continuum of 10 years of a drug's development timeframe. Questions that range from what's the best drug candidate? How does it work? How does it get to the target tissue? What's the right formulation? How do we identify or quantify the efficacy and address the risk and toxicity profile of a drug? These are all questions that today are significantly impacted by modeling and simulation techniques. Outcomes in the end are the key. An outcome that achieves regulatory success at the end of this long investment period of time, that achieves efficacy that accrues to the benefit of the patient, and ultimately our endeavors in terms of the institutions, the companies that we work for, commercial success at the end of the day for that long effort. We've certainly seen in the last year in the COVID environment a tremendous effort by our industry and tremendous success in terms of bringing a vaccine, several vaccines now, to market very quickly that have successfully achieved all of these measurements. And in great part, those endeavors have been contributed to by the modeling and simulation techniques in the marketplace. Simulations Plus, the effort is 20 years in the making. Simulations Plus is 20 years in the making. Founded originally by Walt Woltos back in the mid-90s. The company has grown over the years through the acquisition of several other companies, Cognigen, our PK/PD modeling consulting practice, DILIsym, our QSP, QST organization, and most recently this past year, Lixoft, and bringing the Monolix suite product into the portfolio of the company. We'll talk more about those products and services as we get into the presentation. But today we are an organization of more than 140 employees, global presence. We support the needs of over 250 companies that range from small startup companies to biotech companies, small, medium, and large pharma companies. We support those through software tools and consulting services that we will detail in due time here. Deep expertise, over 75 PhD scientists in the organization with skill sets that range through all of the disciplines of modeling and simulation that serve to support the development, ongoing development and content of our software products, as well as performing support projects for our clients in their endeavors. Next slide. Our vision is to improve health through innovative solutions, a relatively ambitious charge that we work to. Our mission is to create value for our customers by accelerating and reducing the cost of R&D through these innovative science-based software and consulting solutions that optimize treatment options and improve patient lives. This has been our charge for all of these 20 years, and we look forward to continuing this into the future. We operate internally and externally with a set of values. Innovation in science is key. Pursuing novel and creative solutions to positively impact the world is what gets us up in the morning and out of bed and hard at work at early hours here on the West Coast. Respect, promoting a diverse workforce and inclusive culture while serving our communities. Integrity, very important in this world, both internally as we interact as our company, as well as in our engagements out to our clients. Integrity is key in our efforts. And commitment, providing quality products and exceptional services that deliver value to our partners and the people that we serve. Simulations Plus, we pull together all of our products and services to support our clients' modeling and service requirements, be that in the Chem Informatics area, the PBPK area, in QSP, QST solutions, or in Pharmacometric Solutions. Our focus is on science, and our effort is to support your needs in the pharma industry across all of these functions. With that, I'm going to hand it over to Brett to walk us through more detail in terms of our offerings. Brett, you're up.
Thank you, Sean, and Good morning, good evening, good afternoon to everyone who's joined us. We're so glad that you joined us today again. So just to get one layer deeper in terms of what we can provide at Simulations Plus, and more importantly, how we can help. There's two sides to our business. First of all, we have a very robust software business. You can see some of our logos here. You'll hear about those a little bit today in this talk, but more importantly, over the next few hours and the next few hours tomorrow as well. And so we've got products that have been licensed for many years and across companies globally, and have excellent service and development that go along with those products. And then sort of on the lower half of the slide here, you can see our emphasis on consulting services as well. We have consulting services in the various areas that Sean alluded to on the prior slide, and really an expertise in serving customers at all different sizes and in all different contexts. So we'll talk about that more today. And we're not exclusively serving the pharmaceutical industry and the biopharmaceutical industry, but we are in large part focused on that business. And this slide speaks to the fact that our solutions and our products really serve that business from discovery all the way out through approval. And I think that's one of the strong points of partnering with us, is that you can really engage us as a modeling and simulation solution, if you will, all the way through the process. It makes it seamless, makes it, things go a lot more smoothly, and allows for success and connection between the various scientists interacting in these various activities. So we'll start with our Chem Informatics solutions, really our Chem Informatics or machine learning capabilities. Really, the high level point here is to help you find the right druggable candidates. ADMET Predictor, which is our sort of most recognizable platform in this space, quickly and accurately predicts over 140 molecular properties, including things like solubility, log P, pKa, and also sites of CYP metabolism. We've also got an HTPK or high throughput module to help quickly screen a library of compounds on the basis of their likely ADME properties and their potential tox liabilities. And our powerful AI drug discovery tools taken together will help build on your own data to help you rapidly and easily create quality, QSAR, QSPR models. All of these tools, all of our machine learning chem informatic capabilities are really focused on helping you optimize your candidate molecules. What's the right molecule for you, for your particular space and your indication? And of course, we have services and software products in this space. On the PBPK side, physiologically based modeling, both in terms of pharmacokinetics and biopharmaceutics. GastroPlus is our premier PBPK modeling platform. It allows you to do things like predict absorption characteristics based on phys-chem properties, to examine and predict exposure differences across special populations, to predict clinical DDI interactions, and to scale from preclinical data to predict first-in-human dosing. Membrane Plus, another important product for us, highlighted here, enhances GastroPlus capabilities to better predict how fast your drug will pass across really important physiological barriers and membranes. DDD Plus augments our ADMET predictor capabilities by helping you predict dissolution rates and helps accelerate the development of optimal formulations for your drugs. And then sort of accompanying all these things, we have expert PBPK and PBPM consulting services. So if your organization doesn't have the scientist or the expertise to support detailed modeling and simulation plans in these areas, in these product areas and service areas, we can provide that, that help to you. We also have an expert regulatory consulting group led by Sandra Suez-Sharp, who was previously an FDA master reviewer in biopharmaceutics, and she leads our regulatory affairs practice. She really specializes in formulation of pre and non-clinical regulatory strategies. Our group has been helping a lot of folks on the regulatory side, and we'd love to partner with you, really around strategy in that area as well. So now let's move to a few case studies that help highlight how our Chem Informatics and PBPK, PBPM solutions have really helped clients, because that's what we want to focus on here today. Practically, how can we help move through the process and help you with success? So here on slide 17, just want to highlight that with GastroPlus, over 70 different approved drug product applications have been submitted that include simulations with this product. That's an astounding number. You can see some of the name brands here on the slide. But many of those, many of those submissions were also, we learned about through surveys with our customers. They aren't necessarily clear in the labels that GastroPlus has been used for all of those, all 70, but we have sent out surveys and really heard back that GastroPlus has been used in a wide array of areas as well. Some common use cases include widening the dissolution acceptance criteria, demonstration of bioequivalence, supporting CMC changes, DDI assessments, and many more. And so just, to give a few specific examples here as we move forward, this is a poster presented at a prior AAPS meeting that really demonstrated how a GastroPlus PBPK model of a non-engineered formulation was then used to simulate a virtual bioequivalent trial for a particle engineered formulation. So you have a formulation change. You can kind of see that depicted in the table here. We have particle engineered versus the non-engineered formulation, and the two are being compared. And what this allowed us to do in partnership in this particular application was with Janssen, was to simulate that virtual trial, to submit those results to the FDA. And most importantly, the take-home message here is that the FDA accepted these results in lieu of a bioequivalence clinical trial. So there's an excellent example of modeling and simulation saving time, saving money, and helping move a development process along more quickly. Here on slide 19, we have another example of GastroPlus PBPK helping to explain and predict significant food effects for a on-the-market compound called Picray. So these results were submitted with the NDA. First of all, a model was developed that really successfully captured the food effects, and that particular PBPK model, which captured this fairly dramatic fasted versus fed food effect, was then submitted with the NDA as a foundation for future bioequivalent studies, DDI assessments, but also to support the drug label. So we have an example of a model that is moving through with a product through the process, being validated by data that you have today, and then being used to predict scenarios and solutions for the future. Lastly, on the PBPK front, we utilized an updated GastroPlus PBPK model of Tamiflu. This is a model that was originally developed and then validated and sort of updated for adults and pediatrics. And I'll take that as an opportunity to highlight the importance of the fact that pediatrics are included in GastroPlus, and that's an important special population in which PBPK modeling is excellent use case for. And so in this case, that model that was updated and developed with this pediatric and adult data for Tamiflu was then able to support CMC changes and help establish what's known as a safe dissolution space. So the impact, again, we're using data that we have, developing models and validating those models with that data, but then turning our attention to the important questions that we can address with those models that either help support and help augment clinical trials and design, or help in lieu of those things. And this is a great example here of a biopharmaceutics application. So really focused on absorption and formulation and sort of the upstream events. And what we've seen as of late is a push into this area. Here's a recent guidance, an example of a guidance put out by the FDA. And so we can expect more and more of these types of PBPM, or physiologically based biopharmaceutics applications, with this recent guidance. And GastroPlus is the premier platform for this. It's also an excellent platform to use for clinical DDI PK predictions and other clinical applications as well. So now we turn our attention really to mechanistic modeling, and this is typically termed quantitative systems modeling. Some also would refer to it as disease modeling. We have a number of key products and services in this area. On the safety side, we focus on the liver with our DILIsym platform and the kidney with our RENAsym platform. On the pharmacology side, we have focused on metabolic diseases like NAFLD and NASH, on pulmonary diseases like idiopathic pulmonary fibrosis, We've sort of branched into related areas for both of those, and most recently worked quite a bit in heart failure as well. So, in this space, we build platform models, both custom models, new models for our clients, but also use our existing models to help address key mechanistic questions, key questions related to drug target, related to combination therapies, and also help predict the safety of a molecule, going forward. So let's look at a few examples of how this mechanistic modeling can practically help, a development team or a particular product. Here we have an example of a poster that was presented with Genentech, and this poster was focused on the use of NAFLD-SIM. This is our QSP product for NAFLD and NASH drug development. And here we focused on an anti-FGFR1 KLB antibody, and the simulations were first set up and designed so that they would match clinical data that was already in existence. So we put the known sort of mechanisms of action of this drug, of this protein antibody, into our platform. We simulated those mechanisms of action, and then we optimized the model to be in line with some clinical data that was available. And really the key question we're highlighting here is the effect of a particular mechanism, the question of whether a particular mechanism is important or not for a certain outcome. So if you sort of draw your attention to sort of the pink dots here, where adiponectin, as an example, mechanistic effect of this compound, was not included in the simulations. And what we see is that no change in liver fat in these patients in our simulations versus the black dots, which clearly show a change. However, when we include this adiponectin effect in the modeling results, we see, a dramatically different result, and a result that very much matches with the clinical results. And so this is a kind of mechanistic question that you can address with these platforms and help drug development teams understand what interactions or effects their drug may be having, and how that, and then ultimately how that knowledge then translates into future patient types that would be targeted, or future dose regimens that may be optimal for treatment, and also how the drug may work with other therapies. We can kind of shift gears from the pharmacology side into the safety side and highlight a few of our safety case studies. Here we're focused in particularly on DILIsym, our liver safety platform. And in this example, the first example, we actually simulated two compounds head to head. So one is called lixivaptan, the other tolvaptan. These are compounds meant to treat an autosomal dominant polycystic kidney disease known as ADPKD. And what we found was that lixivaptan seemed to differentiate itself from tolvaptan from a safety perspective with our model. And this plot here really, which is called an E-dish plot, really shows that all of the patients simulated with lixivaptan stayed in what's known as the normal range, or the normal quadrant for this drug. Whereas when we ran the comparable sort of therapeutic targeted or on the market, dosing regimen for tolvaptan, we did see patients who were in, the quadrants that suggest liver injury. And so this was actually submitted by Palladia Biosciences, the developer of lixivaptan, in support of their IND, which they submitted, and they were subsequently approved for moving forward, and are currently in clinical studies. So it's an example of a head-to-head comparison of two molecules. And another application here is where we looked at a whole suite of macrolide antibiotics. And so these were drugs that, some of which were on the market. Solithromycin was one that was in development. And the question was, what mechanisms might be responsible for the liver enzyme elevations that you see with some of these compounds? And are they different? Even though the molecules may be structurally similar, do they have different mechanisms, or are they all have the same mechanism? And we were able to do an analysis of the molecules and to show that even though they were structurally similar, they had quite different signatures in terms of what aspects of the what mechanisms each molecule may have caused related to drug-induced liver injury. And those results were actually presented at the advisory committee meeting for solithromycin. Next, we'll highlight an example where we analyzed the compound ubrogepant. So ubrogepant is a CGRP antagonist used for the treatment of migraine. a prior molecule that had been developed, telcagepant, before ubrogepant, had actually shown some serious liver injury. And so the question that we faced was, can you simulate telcagepant and recapitulate the serious issues that were seen? And then if so, can we look forward and predict what we might see with ubrogepant in clinical studies that had not yet been run or completed? And so we, first of all, were able to validate that our predictions for telcagepant were correct. We collected the right in vitro data. We developed a model of exposure. We made those predictions. And yes, we predicted serious issues with telcagepant safety. When we made the predictions for ubrogepant, we saw no such issues. We predicted it to be safe. Subsequently, they took that information along with other information, made the decision to run a phase 3 study. They were successful in seeing safety in that study and also efficacy, and they received approval by the FDA not too long ago. So an example again of a head-to-head comparison of multiple molecules, somewhat chemically similar. The last example on the safety side highlights a drug that we've heard a lot about recently in the news, and that is remdesivir. This is now being used and marketed by Gilead for the treatment of COVID-19. And what we were able to do in this case was really to take the DILIsym platform and analyze a molecule to determine if we could to determine what mechanisms may be responsible for liver injury signals. So there are some liver enzyme elevations that are observed with remdesivir. The question is, what causes those? And in this particular case, what we found was that the mechanisms that we supposed may be causing those were likely not the causes of the liver injury. So sort of an interesting outcome. Nevertheless, important was really on getting into the details of that mechanism with a modeling and simulation tool and helping the developer understand, what may be going on with their compound and what may not be going on at the liver level. So we've talked about our Chem Informatics or machine learning capabilities, our PBPK and PBPM capabilities. Those really feed very seamlessly into our mechanistic modeling capabilities. And now I want to turn our attention to our pharmacometric solutions. And these are especially important, in the clinical development area. We have a number of important software and services to touch on. So first of all, our Monolix platform products provide the ideal cutting edge model development tools to help you efficiently perform your own population PK and PK/PD analyses. But you may have your development team could also collaborate very closely with our expert staff of 50 cognitive pharmacometricians and clinical pharmacologists, and we can really help plan and execute the population-based exposure and exposure response modeling required for regulatory approval. So we have excellent do-it-yourself solutions and partner with us solutions. We also have our PK Plus and PK Analytics platforms to provide standard PK metrics, which quantify drug exposure and have very intuitive graphical user interfaces. So I want to take a few minutes to turn our attention to some specific examples in this area of pharmacometrics. These examples are really good for showing not only how our consultants work with our clients, but also how you can use our tools to do these types of applications in-house as well. So our first example here is really focused on QT prolongation and risk assessment. So we developed, first of all, a model of the pharmacokinetics of Quizartinib and its any major metabolite as well. So we first had a model that recapitulated or described the pharmacokinetics, but then we connected that model to risk from QT prolongation. So we utilized sort of this model of exposure and response, in this case, a risk-based response, to look at alternative dosing regimens, to support labeling, but also in this particular case, to help quantify the risk that would be associated with patients who happen to be on strong CYP3A inhibitors. And so you can really see an example of how we have the connection from building a model that describes the exposure of a compound, connecting that to an effect, and ultimately utilizing that to impact what happens clinically. What doses may be given, and how you're actually treating patients, particularly special populations, like in this case, those on strong CYP3A inhibitors. Second example here is a nice example that ties together sort of our last two sections. So this example focuses on escalabarzapine acetate, a mouthful. For this particular compound, we developed utilized a PBPK model and also a population pharmacometrics model. So we combined more of a statistical model of assessing and then predicting exposure with a physiologically based model. Initially, those models were developed with data from pediatrics aged 2 to 17 years, as well as on data from adults. So we're able to take and construct models, first of all, for, let's say, a regime of ages that was known, we had data for, and then importantly, apply those models for patients between the ages of 1 to 24 months. In this case, the focus was on the treatment of seizures in those very young patients. So we can take a model, or in this case, two models, different types of approaches that can be utilized together to extrapolate from space where we have known information into space where we don't, and the prediction of exposures and optimization of doses for children ages 1 to 24 months is an excellent example of how modeling and simulation allows you to extrapolate and push into those spaces that are very difficult to make clinical assessments of, and important really to have a great understanding of where you're going with dose when you go into that very vulnerable and young population. So here on slide 30, we kind of look at, turn our attention to a monoclonal antibody. This is nivolumab. And this third case study really illustrates the use of model-informed approaches to support understanding of exposure-response relationships and new indications. So first, we refined a previously developed PopPK model for nivolumab. It, by the way, is approved for the treatment of multiple cancer types. And then we used, this to really examine the intrinsic factors that would explain PK variability. And if you look at the different graphics here shown, you can see that there's various factors that were involved in this analysis. So we have things like different infections that were on board, GFR or kidney function, sex, race, tumor type. So there's different covariates that we analyzed. And this is an example of really an evaluation of covariates and how they can have an impact, and that helps you sort of understand and tie back into clinical scenarios and trial design. So here on this on slide 31, this last slide, in terms of practical examples, really want to shift attention to sort of a framework, conceptual framework. And this case study is really focused on the use of model-based clinical trial simulations to de-risk development. So what you're trying to do, and you can kind of focus your attention on the graphic on the right-hand side as sort of a loop here, is quantify the probability of success with planned trial designs and dosing regimens. So what this allows you to do is to say, let's first develop a model and let's simulate different trial designs. You may, look at things like inclusion-exclusion criteria, number of subjects, the dose, the dosing regimen, and frequency, all of these different scenarios. And then you can use that model to simulate the actual trial and then do statistical analyses on those results. And then we want to actually do this in a repeat fashion. So in a loop fashion, we continue to simulate more and more trial designs and analyze those statistically. And what we're trying to do is look at what if scenarios, and ultimately by simulating these various trial designs in advance, we help increase a company's overall chances of positive, clear results and drug approval. We all are hopeful that the drugs that we develop will be effective. The last thing that we want is to develop an effective drug, but then the statistical power of the trial or the design of the trial, the number of subjects, or the type of subjects involved doesn't allow us to really see that efficacy, and ultimately could lead us to a scenario where we don't get approval. So, this iterative, pharmacometric approach is really great for trial design and trial design optimization. Okay, so hopefully, I've given you a brief overview of our products and services, but more importantly, some tangible examples of how we've helped companies and continue to help companies. And I hope that, these very quick and sort of high level overviews has left you wanting more, more case studies, more opportunities, more chances to interact with our scientists and others. And if so, get ready for, the next few hours today and tomorrow. Many of these will be covered in more detail. You'll hear from others on our team, but you'll also hear from many external experts who have invited to the conference and have graciously agreed to participate. So with that, thank you very much for your time. And Sean and I'll be happy to take questions. Arlene.
Thanks, Brett. Thank you, Sean. So we'll give everyone, there's 164 of you here, so I know somebody has a question, a chance to ask a question. Otherwise, we'll assume that what Brett and Sean gave you was completely clear, and now you're ready to get started with the rest of the conference. So while you're formulating your questions, and I'm continuing to look at the Q&A panel, I will remind you that at 12 o'clock, the preclinical, discovery, the model student, and the first in human phase 1 tracks will start with their first speakers. So if we don't have any questions in the next couple of seconds here, then we'll go ahead and let everyone go back to the lobby, review the agenda, and take a minute to bookmark those sessions if you haven't already done so. Again, somebody did DM me a question about, will webinar replays be available? Yes. So in about a week, go ahead and check our resource center on our website, www.simulations-plus.com. And at our resource center, you'll find all of the replays from all of these sessions, both today and tomorrow, including this session and the keynote for tomorrow, Women in Science. I don't see anything else here in the chat or Q&A. So Brett and Sean, great job. And we'll go ahead and let everyone go and get started for the beginning of day 1 of the 2021 MIDD Plus Scientific Conference. Thank you for joining us today.